So moving into module 1.3, we're going to start talking about the fundamentals of genetics. So we've discussed why we need to do crop improvement. We've discussed the germplasm, where we're going to store that germplasm in case if we ever need it again. So we know where to go get the information. What we need now is the understanding of the information so we know which accession that we're looking for when we're considering what new plants that we need. So again, I've mentioned this several times. We took the best of the best and we cultivated those plants so that we have this genetic recombination. We've got this genetic variation, and we know now what the best plants are going to be. And so when I think about this, remember that diagram that I showed you of Teosinte. They just really kind of bred that out. They kind of had an understanding of what they wanted to do with it, but again, it wasn't until the 1900s that we, after Gregor Mendel, that we really understood what these genetic principles are. So just a look at a little bit of the background of genetics, and we've already mentioned, Darwin talking about this natural selection. We have Gregor Mendel, who kind of came up with the basics of the theory of genetics. That's really going to be, he is considered the father of genetics. Next, as we start to, some of these other scientists have, isolated these nucleic acids. We start to identify the chromosomes, and then it really took off in like the 1950s when Watson and Crick actually kind of made a DNA molecule and and kind of replicated that. So, DNA being that double helix, that deoxyribonucleic acid, and they really got that structure down. And once we understood the structure, that really opened the door for understanding where the genes fit and the different ways that we make those genes, or the way that the DNA replicates itself and then makes those genes. So we want to go ahead and talk a little bit about some of the terms that we're going to use in genetics. So we have a trait. And that trait is that characteristic that we're looking for. We have dominant, and this is going to be the trait that is expressed. This is the big A. We have big A and little a. And so recessive is going to be the little a. And previously we talked about alleles. So different forms of that characteristic. Next, we have homozygous and heterozygous. And so homozygous is going to be big A, or little a, little a. So two of the same alleles. Heterozygous is going to be big A, little a. All right, so moving into these nucleic acids, again, I don't really want to get into how we unwind these strings, and when we go through transcription and translation, I really don't feel like that is what we in agriculture are really supposed to be spending a lot of our time on. But we do need to understand that it is these it is this genetics that is these nucleic acids, RNA and DNA, that ultimately give us the information that makes those genes. And so if we understand what the building blocks of that gene are, we can then start to manipulate it. Now, as an agronomist, I'm not going to manipulate those genes. I'm going to ask a plant breeder to do that. So they know more about how that process works. And that plant breeder is going to go back to one of those, the National Plant Germplasm System, and get one of those accessions, because that scientist that works inside of that particular facility understands how to transcribe and translate and make that new gene. So as I mentioned, we're just going to kind of go back a little bit into some of our biology and understand that we're going to have this translation and this transcription, and that, this will unwind and then kind of replicate itself so that we can make more copies of this. And that RNA is going to be the template that we make new DNA with. Actually, it looks like, and also that DNA will give us this RNA. So now we're going to make these proteins and these amino acids from this RNA. So when we have replication, we have these different combinations and these different bonds. And so for this, here you can see that we have cytosine and guanine, and C always bonds with G. And that we have adenine and thymine. And so A always bonds with T. And so here we have, kind of our double helix, our DNA that is always going to bind this way. And the way that those different combinations of the adenine and the thymine, along with that cytosine and guanine, we start getting into when we look back here where we have different combinations. It's not always 2G, 3U's, 2G's. These can change based on these amino acids that are there. And those are going to make up the proteins with inside of that gene that do a specific thing that really cause the allele. So the gene is going to be that nucleotide that encodes a protein. And each one of those triplets is called a codon. So again, this isn't really like a hardcore down to the electron microscope molecular genetics, but understanding that there are going to be this gene that ultimately produces a trait. And so we want to manipulate that gene to express this trait either all the time, or maybe we want to take a gene out. Maybe we want to remove a specific susceptibility to a particular environment. And so this gene, one independent gene, affects our phenotype. So genotype ultimately affects the phenotype. So we seek to the molecular geneticists that are going to quantify the different structures of how that protein is encoded to make that gene. We are going to we're going to rely on them to manipulate the gene to give us the genetic resource in order to influence these particular phenotypes. Okay, so it's important to understand a little bit about cell division. We will talk about meiosis probably more when we discuss fertilization and understanding how these gametes and this different genetic recombination actually occurs. Okay, so we're mostly going to be trying to focus on this meiosis, but mitosis is what happens when just general cell division. So mitosis is going to be for plant growth. Meiosis is going to be for reproduction. So as I mentioned, we just kind of go through these phases real quick. For this particular class, we're not really concerned with mitosis. I remember this as, this is interphase, but this is PMAT. So prophase, metaphase, anaphase, and then telophase. And so we'll go over this again in module 1.3-B because that is going to be meiosis and how we understand those chromosomes. And then when we start talking about fertilization, for moving into plant breeding, that's going to be important there. But mitosis is going to be regular cell division that is more about plant growth than it is about plant reproduction. Now, meiosis, here we have a picture of prophase 1, and where these two different genes or these two chromosomes are now crossing over and creating this genetic recombination. Right? So maybe this is coming back from high school or maybe a biology class that you've taken. Again, we really just need to have an understanding, like a brief understanding of how this process actually works. And we'll go over this in another module. I want to kind of get through the fundamentals and the basics of like chromosomes. What does it mean to be diploid? What does it mean to be haploid? So when we have when we're doing mitosis, we have both sets of those chromosomes. We don't need to recombine with anything else. Those cells have all the genetic material that they need because all they're doing is creating more plant growth. For the sex cells, those are haploid, so 1N. And so plants reproduce the same way that humans do. We know that humans, we need the male donor and the female donor in order, so 1N plus 1N to make 2N. So within those, we have homologous chromosomes, and that is going to be the pair of those chromosomes. And so in the vegetative cells, they've got the same gene that affects the same place or the same trait in the same corresponding position. Now, that is also all combined within the genetic material of that gamete or that sex cell, that 1N haploid. And so, as we mentioned again, we have alleles that they occupy the same position and they affect the same trait. So here we would have this allele or this location or this gene for phenotype A. And this might be, we're going to talk about Mendel's genetics. And so this might be purple flower. So this might actually be big A. And this might actually, so this might be dominant. And we're going to see this expressed as purple. And this might be recessive. And this is going to be the white flowers. So here we are again with our phenotype equals genotype by environment. The phenotype is going to be the actual plant's physical appearance, the chemical composition, how it behaves, how it responds to its environment. But the genotype is the genetic makeup. So this is what we can see. This is what we can't see. So we have our genotype, which is going to be, our big A, little a, big B, big B. But what comes out is actually going to be either a purple plant or a white plant. So we have homozygous. So both of these are big B, and the big B is dominant. So we have heterozygous, where we have big B, little b. Now, the dominant trait will be expressed, but if we were to breed this again with a white flower or a homozygous recessive, we would start to get more of those big B, these white flowers again. So we don't really know what the genetic what the genetic component is just by looking at the plant. So we have to do back crosses and test crosses in order to determine kind of if we succeeded in getting this trait either in or out. And so when I'm thinking about environment, the one thing that comes to mind is going to be hydrangea. And so, this is the phenotype. The genotype is likely to be the same. However, when we have low pH, we get a blue color. When we get a more alkaline pH, we start to get a pink or a reddish kind of color. All right, so that's going to end module 1.3a, just to kind of give you an idea of some of the things that we're going to talk about in the next module. Okay, so in this next module, we're going to talk a little bit about Mendel's experiment and kind of go back and read, kind of, because remember, he's the father of genetics, and it was the basis for understanding that phenotype equals genotype by environment. Just a short introduction. Many of us remember he studied those pea plants, but what's most important is that he came up with these two different laws of inheritance. So the law of segregation and the law of independent assortment. So when we're, he studied these different characteristics and traits of these pea plants, and he found that these were the dominant traits. So these are going to be the big letters. So, big A, like these are the uppercase letters that for the most part, most pods are inflated, or that most of the plants are tall. The recessive traits are going to be like white flowered or wrinkled. I think these are probably the two that he studied the most of and that most experiments demonstrate. So when we're talking about crosses, Mendel performed these different crosses, one being a monohybrid cross. And that is looking at just one particular trait. So just flower color. And so we have this F2 phenotypic ratio, and we'll see this in the next slide, what the difference between the ratios are. So remember that phenotypic is what we actually see. So in this in the next slides, we'll see this monohybrid cross where he was looking at the different flower colors. And when he crossed those, the ratio was 3 purple to 1 white. That is the phenotypic ratio. The genotypic ratio is actually like the letter. So how many big A's, how many dominant flowers, homozygous dominant, how many heterozygous dominant, and then how many homozygous recessive. Next, we have a dihybrid cross. And so now we're looking at 2 characteristics. What if we had purple flowers with round peas crossed with white flowers with wrinkled peas? And I mean, it can get as extensive as you want to, because a lot of these different traits might be affected by different things. Now we start getting into more of a genetics course and what happens when you cross these, and how do these genes interact with each other? Do we always get the same outcome? And how do we test for that? So for the most part, we're just going to focus on the monohybrid and the dihybrid cross. When we do our Punnett squares, we're just going to kind of try to get an idea of what happens. What are the possibilities when we conduct these crosses? So for Mendel's experiment, he took 2 flowers, and so to prevent self-fertilization, he cut the stamens off. So to make sure that we could get the pollen to transfer onto the pistil. So we want to make sure that we're intentionally taking parent 1 and making sure that we're crossing it with parent 2, and that we didn't have parent 1 actually self-fertilizing, and we basically get no genetic recombination. So this is where having that pollen in that germplasm, and we're storing that if we needed to, we could pull that pollen out and then fertilize a different plant. And so, as we've seen before, some of the crosses that he did were for flower color, seed color, and seed shape are going to be the three ones that we talk about in the next few slides. And so, when he crossed these, what we have is the F1 generation. And remember that in a monohybrid cross, we're going to have a 3-to-1 phenotypic ratio. So the F1, the dominant trait, is going to produce at least three of those phenotypic, what we see in the field or for that experiment. So here we are again. We have the parent generation. Excuse me. We've created an F1, and then we'll conduct a back cross and find that three of those plants. This is now our F2 generation, where we have three of those plants that produce a purple flower, but one of those produces a white flower because that recessive allele is still inside of this genotype. We just don't know where it is. And so also think about that this F2 generation, this is really the phenotype. We don't know which one of these flowers has the actual, which one is homozygous dominant and which one is heterozygous dominant. So we'll conduct back crosses, and that's what he did in order to find out which one of these plants actually transferred that genetic material. So he came up with this law of segregation, meaning that each one of those gametes carries one allele of each of those genes. And so this is where we start talking about one in, remember we discussed diploid and haploid. So we know that when this particular generation, these alleles really constitute just one potential genetic recombination. So if we were to cross yellow peas, which are homozygous dominant, with green peas that are homozygous recessive, in our F1 generation, we'll get a heterozygous dominant offspring. And we can have two of these gametes. So now once we have this, we're then going to cross these again. We're going to cross these two, and we now get our F2 generation. And this is where we have our 3-to-1. So our phenotypic ratio is 3-to-1. We have heterozygous for yellow pea, heterozygous dominant for yellow pea, homozygous dominant for yellow pea, heterozygous for yellow pea, and then one homozygous recessive. So the phenotypic ratio, yellow, 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 3-to-1 green pea. But notice the genotypic ratio, remember this is a 1. We have 2 heterozygous dominant and then 1 homozygous recessive. So when we do this monohybrid cross, we will always get this phenotypic ratio of 3-to-1 and our genotypic ratio of 1-to-2-to-1. So from that, what he found was that a gene can have more than one form or allele. And so the offspring will inherit two of those alleles, and it's whether it's going to be both dominant traits, or both of those dominant alleles. Will it be one dominant and one recessive, or will it be two recessives? And so the ultimate goal for plant breeding is to get rid of these recessive traits. We want to always produce homozygous dominant lines. So we know that we get that trait expressed every time. We don't have to worry about a potential green pea popping up because we've removed all of these. So once we perform these back crosses, obviously, if we were trying to make sure that we had all yellow peas, we would take these plants out of the breeding uh, scheme or scenario. We would just automatically remove these plants. Next, we would then back cross them against what we know to be a homozygous dominant plant. Next, we have this law of independent assortment. And so this means that each of those traits, so this is going to be moving into a dihybrid cross, that each one of those traits is independent of one another. Remember that those gametes right here, each of these gametes acts independently. This is our one end. And so we can get multiple combinations of that. So when we do a dihybrid cross, where we're trying to cross two different alleles and see what the outcomes possibly will be, this is why they act independently of each other. And that's why it's called independent assortment. And so remember that this comes out as a 9. So we're like, when we do this, you'll see this in the next slide, this dihybrid cross will be 9, get back, 9 to 3, 3 to 1. Okay, so previously we had this, our parental generation, we have yellow peas that have a round phenotype and actually a genotype as well. And we are going to cross that with a homozygous recessive green pea and a homozygous recessive wrinkled pea. And so what comes out, we'll do this Punnett square, and each one of these will, I guess when I'm thinking about this, I'm going all the way back to the FOIL method, where we will cross this this Y with this Y. So we get big Y, and then this R with that R. Next, we'll cross this Y with this little y, big Y, little y, big R, big R. And when we go over this, as far as for the assignment where we're going to do our own Punnett square, this will be a little bit better. You'll get a better understanding of how to do these different crosses. And so ultimately, this comes out that we have at least 9 round yellow peas. 1, 2, 3, 4, 5, 6, 7, 8, 9. Get back. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 at least are expressing this yellow round phenotype. Now the genotype is not homozygous dominant except for this one. We know that we have 3 green rounds, 1, 2, 3, and that is because we have this these recessive traits are now being expressed through this combination. And then finally down here we have, what is this, 1 yellow wrinkled. Oh no, we have 3 of these yellow wrinkled, and then we have 1 green wrinkled pea. So this is the independent assortment, meaning that each one of these alleles, this big Y, interacts with each one of the Ys along this with this other parent. So this is really like the potential for these different combinations to occur. Another thing that he talked about was this incomplete dominance. And so if we kind of understand, kind of what he did here was, what if we crossed a red flower with a white flower? Would we get a pink one? And if we did, what is the ratio for that? So there is the potential that we get a different genotype when we cross two dominant types of flowers, right? So here we have incomplete dominance because we have our homozygous dominant and our homozygous dominant white flowers. And when we cross those, we get a homozygous pink flower. So just to kind of get an idea of what that looks like as well.